very grateful for not being the first speaker. We've had some introduction to prion diseases and some terminology, which is definitely going to help. So I'm going to start with a slide that in some ways I find quite depressing to show, because I think that slide's been in use for 20 years or so, which is an overview of the mechanism of prion propagation, and that's really the level of detail that we understand this process at the moment. And as Richard's explained, there is a pool of endogenous PRPC, the normal prion protein. It's ubiquitous through mammals, through all tissues, and it does us no harm. But through a process of conformational change to various related isoforms of abnormal PRP, we get PRPSC deposition and ultimately damage. And this is an autocatalytic cascade. Once some prion seeds are present, there is a runaway reaction that leads to large amounts of PRPSC being deposited, and that's the level of understanding that we have, really. So this is showing the sort of organization of post-translational modifications in PRP. So at the top is the prion protein in a linearized form. It has a start, a C-terminus, an end, which then has a GPI anchor added that tethers the prion protein to the outside of a cell membrane. And there are two positions that can be variably glycosylated and have sugar trees attached. And that leads to three glycoforms being visualized by Western blotting, which I feared I might have to introduce, and I don't really. So essentially, if a tissue is homogenized and treated with a powerful proteolytic enzyme, proteinase K, the residual proteins can be separated based on size through a gel and visualized with an antibody. And we can see characteristic banding patterns for abnormal PRP. There are additional bands that appear here and there. So here, if we imagine taking samples of tissue, I'm used to pointing, which is difficult when there's two screens. If we take separate samples of tissue from an individual brain, we might see variable patterns. So on the bottom, the three classic bands associated with PRPSC, but there's also an additional band that's seen variably, an 8 kilodalton band. And it is possible, actually, dependent on sampling, to have tissue samples and homogenates that just contain that 8 kilodalton band and no PRPSC of the sort of archetypal form. And if you take homogenates that just contain that 8 kilodalton band and transmit them to transgenic mice, it doesn't transmit any disease. There is no TSC associated with that band. However, histological examination of the mice will show that the amyloid has been transmitted. So there is PRP amyloid propagating in tissue as well as prions. There's a whole range of sort of abnormal PRP forms, as Richard alluded to. And in terms of diagnostic markers of prion disease, Western blot detection of PRPSC has been around a long time and is a very robust method. Richard, again, has described these things. MRI is very good these days. The range of surrogate markers, not necessarily ideal for diagnosis, but moving forwards as clinical trials may be beginning and new therapeutics emerging, there is a role for biomarkers, and there's a range of other proteins that might have utility, indeed PRP itself. And there are the amplification methods, QUIC and PMCA, both of which are in widespread use in many laboratories, and QUIC is fairly routinely used for diagnosis now. Some of these methods can differentiate between strains. Obviously, Western blotting, PMCA can, and there are some cell culture assays for prion infectivity that can give strain differentiation. I'm going to switch to some hardcore structural biology. And this is an extract of some figures from a paper from many years ago showing the structure of a PRP amyloid fibril. So recombinant PRP is allowed to assemble into fibrils and examined by electron microscopy. So this is an electron micrograph. and segments of those fibrils are extracted electronically and aligned, and that allows you to reconstruct an image of what a PRP amyloid fibril looks like, as shown in the right-hand panel in blue. And so that's what we thought a prion might look like. However, it's not what a prion looks like. They are a quite distinct structure, different from amyloid. So this is ex vivo material. These are purified from an infected rodent brain. And you can see that these structures are paired. There are two fibrils joined together with some material in the center. This is a sort of blow-up. So they're a distinct structure from amyloid. This is a sort of summary of the situation. We've got recombinant PRP amyloid fibril compared to a prion rod, and schematics showing those in the panel on the right. So they're quite different structures. And if we're thinking in terms of some of these amplification methods and what they may be amplifying, then QUIC is probably amplifying from both of these forms, but PMCA replicates from the prion. So just to give you an example of the sort of thing PMCA can do and how it's read out, this is an old example of a PMCA reaction used to differentiate between control and infected blood samples, again from a rodent model. And PMCA is able to amplify the very small amounts of prion material in that blood to a level that's detectable on Western blot. I've summarized some of the attributes of PMCA. It can be very high sensitivity, and it does detect authentic prion infectivity, but it's very time consuming, and it has a requirement for ex vivo substrates. It's also, importantly, not quantitative, and it's really very difficult, if not impossible, to manipulate the substrate for the reaction. So the substrates are usually brain homogenate, made from rodents, which can be of a wild type or transgenic origin. PMCA is summarized here. This is lifted from a paper in the New England Journal of Medicine. So again, there's a pool of PRP-C to which a small amount of seed material, when added, leads to growth and elongation. And then there is a burst of sonication to break up those growing prion rods. And after fragmentation, there is an increase in amplification in the number of seeds. And the reaction goes round and round, amplifying material. Now, a real sea change in PMCA has come about by the ability to replicate prions from recombinant PRP. So this is an example of 10 sequential reactions in this instance, and their analysis by Western blot. So on the left-hand panel, you can see an increasing amount of proteinase K-resistant material, and this is derived from recombinant PRP. So the requirements for ex vivo substrate are falling. There is still a requirement for a brain homogenate extract. In this instance, it's from a mouse which has been engineered not to have a prion protein, and the PRP comes entirely from the recombinant PRP, and that makes the substrate easy to manipulate and allows us to devise new ways of looking at prion replication in vitro. This material is infectious. This bioassay is taken from a different experiment, actually, not the one I've just shown you, but the take-home message is that this material generated from recombinant PRP is infectious and kills all the animals that are inoculated with it. We have an alternative assay for prion infectivity in London. It's called the scrapie cell assay. We've had that for several years. Don't worry about the detail here. The important points are that it's quantitative and it detects prion infectivity. It's not a surrogate. It detects infectivity. And we can use this to look at these PMCA reactions. So again, a serial group of 10 reactions. We look at a scrapie cell assay. The infectious titers shown in the bottom panel. You can see that we're reaching levels which are about 10-fold higher than actually you see in a terminal experimental animal brain. There is a fall off towards the end, but we know what that is. That's an aggregation reaction when we get to very, very high titers. This material can be purified, so we can purify these synthetic prions. Top left panel, we've got a Western blot following purification, and the sort of slightly brown panel alongside it is a silver stain gel. So in the left-hand part, we're using an antibody to just specifically see the PRP, and in the right-hand panel, we're looking at all protein. So you can see that the material after purification is very clean, and in fact, the higher band here is actually the PK that we put in for the purification. And we retain a large proportion of the infectious titer as well. The top right panel is the scrapie cell assay result on assaying that material. We do lose some during purification, but we retain the majority. Underneath that is some mass spectrometry data. It's not really the detail that matters, but we've got to a point where we can put it into mass spec, and the mass spec tells us there's no other contaminating proteins in there. So all well and good, and we're not the first to report or make synthetic prions. Once we got the purified material, though, we were able to look at their architecture and ask the question, well, it's recombinant PRP, it doesn't have sugars, doesn't have a GPA anchor, what's its structure? And the answer is it looks like the ex vivo material. They're paired rods. Probably not the best way to look at them, but you've got handouts, and hopefully you can see, certainly in the bottom right where we're zoomed in, that these are paired fibril assemblies in rods. So having the ability to manipulate the PRP substrate in a PMCA reaction allows us to do a whole range of things that we just couldn't do before. Start to use fluorescence techniques to monitor prion replication and amplification in vitro. So we don't need to wait for a Western blot readout. And that first slide I showed you of the sort of replication cycle for prions, hopefully we can put some more detail on that at last. So very briefly, there is a technique called Förster resonance energy transfer, which allows you to probe proximity of different molecules. So on the top left panel, if you can see the donor and acceptor, if we excite a molecule with green light, shall we say, if it's on its own, it will remain green. If it comes in close proximity to an appropriate acceptor, it'll then become red. The energy will transfer across to the second fluorophore, and we'll get an unaltered fluorescence. And this sort of approach has been used to start to dissect out the mechanism of some of the other, I'm not going to call them prion-like, amyloid replication cycles. And there's a couple of figures lifted there from a recent publication. And this approach is allowing real detail to be put onto some of these mechanisms. So one of the other aspects of FRET is that it's the proximity of the two fluorophores, the donor and the acceptor, that give you that regulate the intensity of the emitted light. So the closer they are, the more energy is transferred. So in this example shown here, the more you'd shift from a blue to a yellow. So these are the sort of techniques we can now apply to PMCA. We can start to look at aggregation in real time and look at the proximity of different parts of the molecule. There's also a technique called anisotropy, and obviously the detail here isn't important, but what matters is that light's polarized. You can filter either horizontal or vertical components, and if we excite a molecule with a horizontal polarized beam of light, if that's a large molecule and doesn't move, the light coming out will remain horizontal. If, on the other hand, it's small and can move quickly, it will depolarize as it spins. It will emit horizontal and vertical components. So we can use that polarization to gain a measure of size, of molecular size. And by combining these different fluorescence readouts, anisotropy and FRET, which are all now possible simultaneously in modern plate readers, given advanced lasers and optics, we can start to put real detail onto that replication mechanism and get quantitative results from PMCA. So to achieve this, we've got a range of cysteine mutants we've made in recombinant PRP. There's 6 of them. There's a backbone model of prion protein here, and cysteine's sort of shown in yellow. A more older version of the talk than I thought I had, but anyway. So this is an approximate Förster radius mapped onto the prion protein. So any fluorophores approaching that radius will start to transfer their energy across. So you can see that in the context of that molecule assembling into a fibril, there's plenty of scope for the two to get close enough. So the question marks really are whether we have enough synthetic prions in a tube to be able to monitor bifluorescence, and the straightforward answer is yes. Standard fluorescence can get below a picomolar, although you have to be doing well. Detection limit of FRET's about 1,000-fold higher, and anisotropy is even less sensitive. However, the concentrations of synthetic prions we're getting up towards are about 500 nanomolar, so we are 100-fold over the concentration we need to be able to do these things. I'm not presenting any data on having done it, because obviously it's early days. We're here just 3 months after starting this, but we'll be able to update you in due course. So in summary, well, we can make synthetic prions at very high concentrations and high infectious titers. We've now got a whole range of genetically modified PRPs, several of which are made and refolded. We can tag these very specifically by having these cysteine residues. That allows us to put the fluorescence tags in very specific locations. And we've got the equipment to be able to monitor a whole range of different fluorescence measures simultaneously during the prion replication cycle. So we don't know exactly what's going to emerge from this, but we would expect that there might be variations in the growth rate. There may be different endpoint levels. There might be different proportions of intermediate steps that have been alluded to. But we should now be able to visualize these things and start to get a more detailed handle on that replication cycle. So I'd just like to finish by thanking everyone who's funded this particular project, and to thank you for allowing me to come here today.